Hi everyone. So today in this video we will be discussing this paper, that is single cell multimodal analysis, which reveals tumor microenvironment predictive of treatment response in non-small cell lung cancer. So here this paper has been published by Zheng et al. And it is published in Science Advance 2015. Well, the main if you see the abstract, this paper talks about the non-small cell lung cancer. This paper has been published in Science Advance in May 2024 to 2025 by Zheng et al. And the laboratory of Olivier Gevaert at Stanford University, which represents a major advancement in precision oncology. Well, this study leverages an integrated approach combining multiple multiplex immunofluorescence and H&E histopathology and other advanced techniques like RNA sequencing to characterize a spatial architecture of the tumor microenvironment, and that predicts the patient response to immune therapy. And the basic core methodology what they have used is the NucSegAI model. It's a central innovation. This is the central innovation of this paper, which is the development of NucSegAI, a deep learning model for automated nuclear segmentation and cell classification in histology images. So they have employed a training strategies unlike typical models trained on manual human annotations, which are prone to error. This AI model, it was trained on a large scale H&E staining images, and they are paired with immunofluorescence data. And this allowed the model to use the objective protein expression. For example, they have used from 33 different biomarkers, and which are like ground truth to teach this AI how to recognize specific cell types in standard H&E stains. So we'll be going through all these different figures one by one. So the key finding, what they have found here is it's about the spatial tumor microenvironment, and this study identified a specific neighborhood recurring spatial architecture that were highly prognostic in clinical outcomes. For example, they have used lymphocyte uh, enriched neighborhoods, and they also found they are associated with extended progression-free survival, and they can respond to immunotherapy. They have responders to immunotherapy, and they had a higher proportion of these neighbors. So if we see this figure specifically, figure one, so here they have designed a study using the NucSegAI framework, and this figure acts as a blueprint for the entire study. For example, if we talk about the cohort, it visually summarizes total 115 NSCLC patients, which shows the distribution of H&E slides, which they have named it as multiplex immunofluorescence and RNA-seq data. And the typical workflow, it depicts the NucSegAI training pipeline that is specifically showing how this IF biomarkers were used to label cells in corresponding H&E images. And the goal was to establish the objective, converting a standard flat pathology slide into high-dimensional map of 45 million individual cells. So overall, if we go to the figure two, where they have used the validation, done the validation and performance of NucSegAI, and this figures proves that the AI can actually see what it claims to see. For example, they have utilized the accuracy benchmark. It compares NucSegAI cell classification based on H&E alone and against the actual protein expression in MIF. And visual, if they have also done the visual comparison, usually contains side by side images where the left is an H&E crop and the right is the AI's predicted cell mask for in terms of coloring tumor cells, T cells, blue and T cells like as a blue and etc. And they have also utilized some statistical parameters, metrics like a precision recall curve or even confusion matrices, and they have showed that the model is highly reliable at distinguishing between epithelial cells and various immune cell types. So that's all about this figure 2. And then they have this figure 3, where they have shown the mapping of the spatial landscape, that is cellular neighborhoods. And this is where the biology gets interesting. For example, they have, instead of looking at single cells, they have looked at communities, for example, the spatial clusters. And the figure uses a spatial analysis, often k nearest neighbors to KNN, which we often refer as, to identify 10 to 12 distinct neighborhoods. Example, the immune desert, or the myeloid rich zone, or the lymphocyte tumor interface. And they have also performed the proximity heat maps. It shows which cell like hang out together. For example, they have highlighted how these dendritic cells and CD8 positive T cells are more frequently touching in certain patient groups. And in figure 4, further, they have examined the tumor microenvironment, which features linked to different treatment response. And this figure connects the spatial data to the patient's clinical reality. And they have performed the analysis between responder and non-responder. They have also shown some typical bar charts or dot plots comparing the density of a specific neighborhood between patients with improved on immunotherapy and those who did not. And the neighborhood shift that illustrates the responders have significantly more lymphocyte-enriched neighborhoods, while the non-responders, they are dominant by macrophage-enriched spatial patterns. And they have also shown some Kaplan-Meier curves, and which is the classic survival curves, having shown the patients with high spatial cytotoxic T lymphocyte, their scores have much longer progression-free survival. Finally, we have another main text figure, that is figure 5, where they have performed multimodal fusion and predictive power analysis. And this is the grand finale for the figure that proves the fusion. Here you can see that the fusion, in terms of the ROC curve, this is the most critical part of the paper. It plots the AOC, area under curve, for, as you see, they have shown different kind of values. And the fusion score is almost like more than 90%, showing that the combining all three provides the most perfect prediction. And they have also shown the biological correlation in terms of the heat map or even the correlation plot, showing uh, how this AI is H&E based feature aligning perfectly with the hot or cold signature found in the RNA-seq data. So if we go back to our abstract, so overall, they have shown this. For example, here they have written this: non-small cell lung cancer constitutes over 80% of lung cancer and remains a leading cause of cancer mortality. Of course, this sets the background for the research. And the real problem is, despite the advancement of immune checkpoint inhibitors, their efficacy is limited to 27 to 45%. Even though we have several checkpoint inhibitors, their efficacy is low. That's what they are saying. And then therefore, now their the real problem what they have found is the identifying likely treatment responder is essential and for optimizing healthcare and improving quality of life. So basically, it is quite important to identify the treatment responders. So therefore, what they have done is they have generated multiplex immunofluorescence images. So they created a set of images, immunofluorescence images, and the histopathology and RNA-seq data from human non-small cell lung cancer tissue. And through the analysis of this multiplex immunofluorescence images, they have characterized the spatial organization, like in the same tissue, how they are organized, like around 1.5 million cells based on the expression level of 33 different markers. And to enable this large-scale characterization of tumor microenvironment, they have developed a model, that is NucSegAI, which is a deep learning model for automated nuclear segmentation and cellular characterization in histology images. With this model, they have analyzed the morphological, textural, and topological phenotypes of 45.6 million cells across 119 whole slide images. And through the unsupervised phenotype discovery, they have finally identified a specific lymphocyte phenotypes predictive of immunotherapy response. So they have found that this lymphocyte phenotype, which is highly enriched, due to this immunotherapy response, and that can be used as a identification for which is specific to the immunotherapy response. And their finding has a capacity to improve patient stratification and which can guide selection of effective therapeutic regimens.